Hello, my name is Ivona Cetinić, and I'm an oceanographer at NASA Goddard Space Flight Center. I'm happy to be here today and tell you a little bit about how large field campaigns are match made in heaven with optics and biogeochemistry. This talk is going to be really focused on the science that has been done under experts, North Atlantic Bloom experiment, a little bit of names, and I'm grateful for the teams that have done such a great science, and also for the discussion with some individuals from these projects that helped me outline this talk. Every science starts with a question, and the question can be teeny tiny, or it can be really gigantic. And when we find ourselves trying to understand the oceanic carbon cycle, specifically pertaining to the functioning of the oceanic carbon pump, we find ourselves very often in the realm of large questions, large complex questions. These are the processes that are very interconnected and very poorly, in some certain ways, understood. So one of these large questions actually propelled exports campaign. And without going into details, which you probably know because you heard Dave talk about a lot, exports is focusing on capabilities of us predicting the exports, fates, and carbon cycle impacts of oceanic net primary production from satellite and other observations. And what I'm going to do, I'm going to use this table from the exports implementation plan to kind of lead you towards the idea of the large field campaigns and optics. So these science questions kind of inform the approaches that we want to take. These are the overarching ideas, science, things that we want to do in order to answer these questions. And these approaches that we want to take inform furthermore deployment requirements. Deployment requirements are things such as we want to have a ship moving Lagrangian space to understand the submesoscale dynamics, and then we want to have another ship or gliders to extrapolate these things to the mesoscale. We want to be able to follow these things as they sink through the water column, this carbon, and so on and so on. And you can take all these deployment requirements, start thinking about them in some time and space. And that's kind of how I think about things. I usually revert to diagrams such as this one that you see, where different biogeochemical phenomena or physical phenomena are overlaid on a different time and spatial scales. And so I took these deployment requirements from the experts and put them on top of these phenomena. And you can see that the range of the spatial and temporal scales that experts wants to study on is gigantic. And this cannot be done by one ship. This cannot be done by two ships. This can be just done the army and army of platforms using very complex tools and very specific observations. And this is where we come to the idea of a large field campaign. Large field campaign do have a definition, and the one that I found in NCAR says that these are the campaigns that require significant monetary resources, use several observing platforms, involve multiple agencies, and entail difficult deployments, often overseas. And if this is a logistics presentation, I can talk about it a lot. But I agree with this definition. However, when it comes to the actual science, large field campaigns usually are about answering highly complex scientific questions by using appropriate, sometimes redundant tools, techniques, or methods, and doing it all on a proper, appropriate temporal and spatial scales. So once you have defined that you really need this large field campaign, you kind of know your deployment requirements, you can start moving into the field of what are you actually going to be measuring in order to satisfy the approaches that you want to use in order to answer the questions that you had in hand. And you can start thinking about different biogeochemical measurements of stocks or processes of conversions that you can then be doing on the ship. And then you start thinking about autonomous vehicles that you can add in order to extrapolate and give some kind of spatial context to this measurement. And you think about taking it all the way to the satellite and models. And while all these things kind of build up on each other, they're kind of discrepant in their time-space continuum. And we need a connecting tissue for that. And when you start thinking about it, the connected tissue is actually optics. Optics are a suite of tools that is currently looked at as two groups. We have classical optical tools and imaging tools. And one thing that is connected to them is that they can measure or infer biogeochemical measurements in their very high frequency, which means they take that single point measurement of parse filtering, phytoplankton or DNA or something else, and they're able to extrapolate it to the time and space. They take the information that is important and increase its frequency, either directly, as imaging tools do, And they offer us the single particle measurement, single particle information about the particle size, taxonomy, morphology, or through combining physical measurements of the properties of the light with the biogeochemical measurements in order to develop proxies that can then be extrapolated to time and space. These optical tools are usually the one that we refer to as inherent optical properties, and they can give us an idea of the bulk information about particle size, abundance, composition, morphology, something about concentration type of dissolved organic matter, even all the way to the nutrient concentration. But we can also use radiometry to infer availability of the light in the ocean, and also build really good connection to remote sensing with our biogeochemical parameters of interest. So why do we want to use optics? As I said, these are high frequency tools. These tools allow us to really look at the biogeochemical properties of interest through space and time, used either in a ship or autonomous vehicles. Just as an example of comparison, in the last North Atlantic exports campaign, one of the ships collected an impressive 600 HPLC samples, which is a lot, 600 pigment samples. When you compare it to 2.5 million absorption spectra, they can, in theory, when combined with these pigment samples, give us similar information. It's pretty much nothing. Optical tools allow us to connect biogeochemical measurements of these stocks and processes with Earth observing fleet, give us a synoptic view of the ocean biogeochemistry. And ultimately, these optical tools allow us to put biogeochemical measurements into the scales that are needed for the either small ecosystem models or large Earth system models. These three topics are also going to be touched upon by the colleagues. They're also talking in the optics session, which is Angel, Joe, and Blake, so go check out their talks as well. But they can talk about those things. I'm going to take you back in a realm of why optics in large field campaigns. And here I'm going to use the citation from one of my favorite songs by Daft Punk. It says, called Harder, Better, Faster, Stronger. And through some examples later on, I'm going to show you how optics can really help us to do biogeochemistry better in a proper time and spatial scales. We can do it faster and we can do it stronger. Not only gain what we're looking for, but actually get additional information, a completely different view of the processes that we want to study, or we didn't know that we want to study. But ultimately, the development of these optical tools is harder. You're dealing with two different sets of measurements, two different uncertainties, usually very, very variable methodology. And this is where also large field campaigns come in hand. I'm going to show you one single example that is near and dear to my heart, and that's the one that has to do with backscattering. Backscattering is one of the simplest, backscattering in the form that we're talking about here is one of the simplest oceanic measurement and relies on the principle that light comes from the light source, interacts with something, and comes back towards the detector. If there are things in the water, it's going to interact with it and scatter back, and then we can measure that scatter. And when you think about things in the ocean that can scatter, these are particles, and particles in the ocean, especially open ocean, contain carbon. So backscattering is a really great tool to tell us something about the abundance of the particles, concentration of the carbon, phytoplankton carbon, any carbon that you want. It's a great tool for biogeochemistry because it has a strong relationship with remote sensing reflectance. And it's also tightly connected with the LiDAR-based measurements of the backscattering. But ultimately, the minimization of the technology when it comes to the backscattering allowed these instruments to be present everywhere, on all the autonomous tools, on moorings and all the ships, and increases the impact that this optical measurement can do in order to understand the global biogeochemistry. So how do we build one of these proxies? Well, when it comes to large field campaigns, you don't have a single measurement, as I previously said. There's lots of redundancy in measurements. And here you can see 10 different profiles, all done by the backscattering instrument during the North Pacific exports campaign. And in this paper, Zach Erickson tries to kind of figure out a way to make them all speak the same language. You can see right away that the brown profile here is really different than the rest of the profiles. But if you look at the bottom of these other profiles, you can see some discrepancies. And you can go and then play and build this really elaborate network of relationship between all these instruments and trying to understand where the differences come from, and how to compensate for these differences to ultimately gain something that looks like this, aligned backscattering sensors for the whole campaign. So all the backscattering sensors are talking the same talk. So all our instruments are seeing the same thing. Through this exercise, we realized that majority of this variability and differences are actually associated with the calibration factor, and that differences in calibration factor can lead up to the 10% uncertainty. And that's an important thing, because uncertainties is something that optics can bring to biogeochemistry, and it's often missing when it comes to implementation of the biogeochemical measurements to models or remote sensing algorithms. Then we can take that backscattering aligned measurements and start pairing them up with the particular organic carbon measurements that have been collected aboard the ships. But lo and behold, since we also measured particular organic carbon in so many different ways, we found that these methods also don't agree. Not even that, sometimes the same method and two different ships don't agree as well. So the POC group led by Jason Graff has gone through this reconciliation as well. And following 3 years of work, they came to this relationship. So however simple it looks, it wasn't simple to make. And having done this in a large field campaigns where we can differentiate the natural from methodological variability was really important, because now when we apply this optical proxies in all our tools, it's going to be easier to actually use it to study biogeochemistry. How powerful this conversion is, it's really powerful, because this is an example from the North Atlantic Bloom experiment, 300 samples of the particular organic carbon through the backscattering allow us extension to the 3 million samples collected across the whole fleet of the vehicles that were present in this campaign. Additionally, what this approach gives us increases our confidence in our numbers. Furthermore, it does take a long time, but ultimately is worth it, as you're going to see in some examples that I'm going to give later. I don't want to say that everything is bad and everything is ugly, but it takes a long time to develop proper proxies and proper connection between the optics and biogeochemistry, and it's not that simple. So it's a couple of reviews that have been published recently do really, really good view of how we can use these biogeochemical tools, how we can use these optics tools to infer the biogeochemical processes. And I would really refer you to the paper wrote by Sarah Goering that is really evaluating differences between the techniques that we can use to estimate fluxes in the ocean and how different they might be. But ultimately, can we get biogeochemical closure with optics? When I invented that, they was like, hey, what is that? Biogeochemical closure in my head is like, can we measure a majority of these processes outlined here as being important to biological pump using only these optical proxies and imaging tools, of course, in combination with the discrete measurements done on ship. But once again, seizing the opportunity that we have optics and actually exporting to the scales of the interest. I'm going to take you through a couple of examples that are really constrained within these large field campaigns. I know there's many, many out there, but I really wanted to show the power of combination of the ship, several different platforms, and the proximity of true biogeochemical measurement and optics, and then evaluation of that one on a time-space continuum. Okay, so I'm going to take you to the biological carbon pump, and we're going to start at the surface with the primary production and the composition of the little buggers living out there. And since this is my talk, I'm going to do some little bit of self-promotion. This is the work that I have done some time ago as part of the North Atlantic Bloom campaign, in which we combined two simple measurements of the optics, chlorophorescence and backscattering, and found that this ratio, these two, works really well to determine differences in phytoplankton community that we have encountered in North Atlantic spring bloom. The changes in chlorophorescence to backscattering ratio were indicative of the changes in community composition. And since all our platforms had these two measurements, we could actually extrapolate this now inferred knowledge about the phytoplankton community structure and to start evaluating differences in community over community evolution throughout the spring bloom over different time and spatial scales. Similar approach was done during the NAMES campaign, but in this case, they combined aircraft and ship, which I think is pretty cool. And they used in situ observation of the phytoplankton community collected by one of the imaging tools, IFCB. And they saw as they were crossing this eddy here that there are differences in phytoplankton community on the outside and inside of the eddy. And when they looked at the data from the overfly from the aircraft and the high spectral resolution LiDAR measurements, they saw that there are certain trends that they have observed. They calculated chlorophyll from the KD and the backscattering, look at the ratio, and similar to what we observed in North Atlantic bloom experiment, they saw a change with this transition to different phytoplankton community composition. Furthermore, LiDAR has a capability of measuring depolarization ratio. So they looked at the ratio of depolarization with the backscattering and saw even stronger changes in this index, indicating furthermore that the shift towards the dinoflagellate in the center of the eddy can be seen both by depolarization ratio, chlorophyll, and backscattering that can be measured or inferred from the LiDAR. This is really cool because not only affects this field campaign, but it really informs the future development of the remote sensing tools. LiDAR has capability to see below the surface, and you can see below the surface, see community composition. It has a really, really big impact on our understanding of the biogeochemical processes in the ocean. Now a little bit to the rates measurements. Also during the North Atlantic bloom experiment, Briggs et al. Combined measurements from the ship and autonomous measurements to say something about the formation of the aggregates and timing of the exports in this North Atlantic bloom they encountered. He used a method where from the backscattering, he looked at the variance over mean ratio and connected it to the size of the aggregate formation. And here you can see that the aggregate formation is really timed with the largest amount of exports that he also calculated from the gliders. But when he went and calculated gross primary productivity, net primary productivity by combining ship-based measurements and light and chlorophyll from the autonomous vehicles, he saw two different peaks in this productivity. And further, the second one was actually driven by the physiological changes impacted by the silica depletion in the area. I'm going to take you from my favorite phytoplankton into the things that eat my favorite phytoplankton. This is the secondary producers and secondary production. This is a really recent, not yet published study from Exports North Pacific, where Debbie Steinberg and her team look at the crazy, ephemeral, and completely underappreciated salps and their role in the export in the North Pacific region. What they did here on the left-hand side is they presented the information about paired Mach-Ness net toes, where they found different distributions of the salps for three different epochs of the exports. And they saw different distributions, of course, during the night and during the day. But since this is only six profiles, they give them really good ideas of a heterogeneity of the distribution of these patches of salps. So they looked at the UVP and they saw that the imaging information from the UVP sensor that was deployed about the CTD gives a very similar distribution of the salps. So therefore, now we have Mach-Ness. It was done 6 times, can imply the information, can start using the information from the UVP that was deployed every time a CTD was done. And in combination, the UVP information in combination with some additional autonomous vehicles allow them to map the distribution of the salp population over the area where North Pacific experiment was happening. Furthermore, if you go into the realm of exports, they used, once again, UVP information combined with some fecal production data and also data from the sediment traps deployed aboard the neutrally buoyant sediment trap platforms to infer the production of the fecal pellet. And managed to really talk about both active and passive roles that salt poops have on the export in this region. As I said, this paper is under review, and a couple other papers that were associated with it as well. When it comes to the exports, there's one thing that is really unavoidable to talk about, and these are spikes. I'm very excited every time I see spikes because I know something is happening. It's either zooplankton coming or the aggregates or phytoplanktons being exported. And in this paper, Briggs et al. Used the tools they had in hand during the North Atlantic bloom experiment to infer the carbon export as the bloom was collapsing from these spikes. They used optical tools to separate the spikes, convert them in carbon, and actually observe this export horizon as it was happening through space and time using autonomous vehicles. This export was additionally confirmed with the sediment traps that were deployed in the region. Ultimately, if we want to look at the physical transport that is associated with the biological carbon pump, we can look at the beautiful paper written by Melissa Amand, once again using the data from North Atlantic bloom experiment, where the profiles collected from the glider on the edge of the eddy, they observed these bumps, not only in the physical properties, but also in the measurements of the optically derived particular organic carbon, as well as oxygen. And what they've seen is that based on these patterns and distribution in combination with some modeling, there's a big contribution of this eddy-driven subduction to the biological carbon pump export in this region, and that is not really just localized to this area, but they managed to model it globally. So these are like lots of examples in one space and time, and I wish that I could have given you more. There's many new techniques that people are developing, really exciting things on the export, but I really wanted to give you a complete story where we combine optical measurements with the biogeochemistry in a large campaign and then extrapolate to make a bigger conclusion. I see large field campaigns that are focusing on this combination and biogeochemistry and optics as incubator for the ideas. They're testing new things and pushing the boundaries. And then this science is going to be driving the development of the technology, and this technology is going to be driving the development of the science. For example, this paper that I was talking about that Debbie has written are showing really good usage of these UVP sensors, these imaging sensors that can now be deployed aboard autonomous vehicles. And examples such as this one are crucial for developing the optical proxies, but also application of these proxies to the long-term programs, new autonomous fleets, or even supporting, development, extending the presence of the satellite data in the oceanic realm. With the new funding that is going towards Go BGC and Go Ship and so on, we can see the power of these proxies on much, much larger scales. Additionally, these large field campaigns, they combine optics and biogeochemistry. We see that with each one we learn more. In my timeline, I'm going to start from North Atlantic bloom experiment and then follow them on by names. And then you have exports and comics and many different things happening at the same time. And with each new one, you learn more because the technology the methods that we have developed, the science that we have discovered, answered some questions, started new ones. We took the information, put it in the models that informed certain new hypotheses and pushed us back on the field. But ultimately, it's really great to see these large field campaigns working together because that not allows only for the intercalibration of methodology and similar thing, but also allows for the largest scientific footprint and ultimately a UN endorsement as Jetsons has got it. So this was really fast on lots of things, but I hope you got the gift that large field campaigns are crucial for continuation of our exploration of the oceanic biogeochemistry. Thinking back 12 years ago when I was in my first ocean biogeochemistry workshop, this whole idea of using optics to study biogeochemistry was even completely crazy. But it's great to see how the field has moved and embraced this all. So it's really good to also see that the funding agency keep on supporting these large field campaigns because they are truly incubator for new science. Thank you so much for your attention.